Good morning, everyone. Welcome to the ACOM seminar this week. It's my great pleasure to introduce our speaker today, Alex Guenther. Dr. Guenther is a professor in the Earth System Science Department at the University of California, Irvine. Dr. Guenther research focused on characterizing and modeling the process that control the release and uptake of reactive trace gases and particles and their impacts on the atmosphere. He received a PhD from Washington State University and served as the Biosphere Atmosphere section head of the ACD of NCAR for several decades. He's a fellow of the AAAS and AGU, and he has led multidisciplinary field studies across 6 continents to investigate processes controlling atmospheric composition and their influence on climate and air quality. He also leads efforts to incorporate the results into biogenic emission models that are widely used by the scientific community and regulatory agencies. In today's seminar, he will talk about biogenic emissions in a warming world. And with that, I will leave the floor to Alex.
Well, thank you, Ching. It's really a pleasure to be back here at NCAR. It's been too long. I promise I won't stay away as I have been, and we'll get back here more often. But I really appreciate this opportunity, Ching, to give this seminar, and thanks to Louisa for getting me here to Boulder this week. Really happy to be here and catch up with my friends. So today I'm going to be talking about some of the work that we've been doing and looking at how we think biogenic emissions will be changing in the future. And going to start off with going back to the past. About 3 decades ago, 1991 was a key point for these biogenic emissions research. Previous to this time, back in the 1980s, people were pretty skeptical that these biogenic VOC could have any kind of an impact on air pollution. And it's kind of the standard EPA mentality was like, no way, they really can't be doing anything. But after decades of trying to do something about ozone, it was recognized, and this NRC report was really key for that. And so this was 1991, and it really changed EPA's thinking. They started funding research in this, including a study looking at, well, hey, if these BVOC are important, we know there's all this CO2 getting into the atmosphere. How's that going to impact things? And so EPA had funded this study by Dave Turner and colleagues, and they looked at this and said, well, you know, it's warmer. We know these things are sensitive to temperature, more CO2 in the atmosphere. You're going to have more foliage. Plants like to take up CO2 and make things out of that. They also looked at dynamic vegetation models. So people were starting to do dynamic vegetation models there, saw how climate would change things. And they said, oh, in these models, we see more rainforests. So globally, there'll be more emission. So with those first 2, I think those really are effects. The last one, little uncertain if that's really going to happen. Probably not. But we do think that changing vegetation, natural vegetation, is an important thing. We just still really don't understand it very well. But also the estimate they came up with was about 25% for a doubling. So yeah, not insignificant, but not really huge either. So a fairly small increase. Couple decades later, EPA was still funding this kind of research at a more specific local scale. This was a part of a STAR program. They funded 5 different consortiums to do some modeling work. Myself here at NCAR, we were involved in one of those, and then there were 4 other groups. And all of them used different climate models, different vegetation changing, different but somewhat similar BVOC models. And so what you're looking at here is just the BVOC part. Of course, they were estimating ozone and particle changes, but here just the BVOC emissions. And so what you can see, the red is increases and the greenish, yellowish are decreases. And these maps don't look all that similar. There are some similarities. The southeast is generally redder, increasing more. You can look on the west coast there. One model has the northwest decreasing and southwest increasing, it's flipped for the other. Another model says nothing happening. So they all agree that there were changes. They all actually had a net increase for the US, but regionally, really different things going on. And also these models are not so different, right? I mean, partly we didn't know all that much. So that's 15 years ago. More recently, there's been suggestions, some European groups saying, look, we know there's these other things going on, and one of the key ones is the suppression of isoprene with increasing CO2. So we don't understand the mechanisms very well, but we know that plants exposed to high CO2 will emit less isoprene. And so that's a known effect, but how much of an effect is uncertain. They suggest that it's quite a bit. They did these simulations, so you're seeing over here on the right, they're going from 1900 to 2100, so we're right here, a little bit to the right of center there. And saying, yeah, climate warming is going to increase these emissions, but CO2 is going to balance that out. It's going to be a wash. Land cover has been a big deal. There's been big decreases, the model says, but not so much in the future. But still, their conclusion here and their highlight is a future global increase in BVOC emissions is improbable. So saying that it's not likely that this really is going to be a thing. Looking even more recently, the CMIP-6, comparing all these global chemistry models, chemistry climate models, and there's a plot in there in the supplementary that shows the BVOC emissions, and shows it for 5 models. There are a bunch of other models, but mostly those models have prescribed emissions, so they're not changing. Or they're changing in some prescribed way, or they're using these similar approaches. Even with these 5, there's really 3 of these 5 are using Megan 2.1, so the same model, although, well, and 2 of them are CLM Megan 2.1, so exactly the same model, but they use different drivers and temperatures and other things. One of them is using an older model that we developed here back in the 90s. And then only this one on the top is really different and has more of some of the that European, the kind of European perspective. And you see more decreases here and not so much of an increase. But the others are actually all pretty similar. But again, even with similar models, you can get a fair bit of difference. On the right, you're seeing the increase, but it's mostly increase in most of these models. But it's just partly to say that then still what's in the models that are doing these climate studies is not so much, as our view of 20 years ago or so for the most part. So went through and was looking at studies, so lots of global models. People can run these, publish papers on how things are changing in their model and looking into the future. Looked about 20 papers where people did this and with their models that are doing different things. And you can kind of come up with, well, there's two main things changing. One's CO2 and other greenhouse gases, and then the other is land cover is changing, right? So those are things we're doing to the planet. CO2 does a number of different things, though. So there's this isoprene inhibition, where you decrease isoprene at high CO2. And there are 2 main algorithms that have been incorporated into these models. One would give you about a -3% per decade change. The other would give you about -6%. But still factor a 2 difference, but enough of a difference to make that where you can get down to say there's no change. If you have this -6% and then the others doing +6%, you get nothing. There's the foliage amount, CO2, making more foliage, more leaf area, affecting the type of vegetation, both the CO2 and the warming is going to do that. And then there's this temperature effect. So as it's warmer, you're going to have more of these emissions, CO2 as well as other greenhouse gases. For that, these various studies come up with something of about plus 2% to 7% a decade. So come to range in that area there. For the land use, you can get a lot of different things depending on your dynamic vegetation model and your prediction of what's going on. But for the future, and the idea is in the past, it was probably much greater than this, but a decrease as we went deforested and have more agriculture. But now anywhere from about -4 to +4%, which means the total that you can get is anywhere from about -8% to +8%. So I'm going to go through each one of these and kind of see what we know and what we've been learning about it and see how this picture changes or not. So starting with that CO2. If we go back at 2009, these were kind of the two main studies that were done. There were other studies that said, yeah, this is an effect, but these were the two that did a lot of measurements at different growth environments, and algorithms were developed that were put into global models. So one of those, working with Colette Hield, put it into Megan 2.1 in CLM. And from that, you get about -3% per decade. This other study, which you can actually see in this on the right, this is that curve from that study. And so you can see it kind of doesn't look so different when you look on this scale, but it's different enough, actually, as you go from here to here, you get going, you can get -6% instead of 3%. Part of it is this other algorithm is really kind of skewed by this stuff that was going on in paleoclimate times, 200 ppm CO2, which is maybe not so relevant for what's happening now. So we focus more on what was going on in this range right here. And also this study was based on more types of plants, whereas the other study was one species. More recently, people like Ulu Ninemit's looking at this and also starting to work out what is that mechanism that's going on. It's still a bit of a mystery, but they're learning more about it. They understand the biochemistry and physiology of it. They have more carbon, there's more CO2, you'd think more isoprene, but they shunt it to other things. But anyway, the point with this on the far right there is these are different plant species, really different effects, right? So yeah, if you just pick one plant species and study that, you're going to get something different. So at least a factor of 2. We need to understand these plant-to-plant differences. But another recent finding is looking at, well, it matters what temperature you're at when you look at this CO2 inhibition. And so if you do plants at 25 C, which those other studies, including the one that was done here at University of Colorado and Russ Monson's lab, 25 C is a nice, good temperature for a plant, so that's where those studies were done. Well, it turns out as things get warmer, there's going to be less of an effect. So this is from Tom Sharkey's group, and they looked at this, go 60% reduction with a doubling of CO2 at 25 degrees, drops to 40% at 30, and 20% at 35. So as the world's warming, we can expect less of this CO2 inhibition. We still need to quantify this and get it into the models, but it's something that we're doing now. But it suggests that this isoprene inhibition by CO2, it's a real thing. We need to account for it, but it's probably not such a big a deal as those who've used this to say that there's going to be no changes. So the other thing is the land cover. So the land cover is changing, and in two ways. One is sort of the natural vegetation change, and then you have us going in and making the changes. As far as the natural vegetation change, the studies previously that had been done were all, well, we run our global model with these PFTs, and we see these changes. But of course, they don't really know what the emission is of each of those PFTs. We assign something in the model, but we're not confident about how that changes. And if you have climate change, a temperate broadleaf forest could completely change its emission, even though you have more temperate broadleaf forest. And so we really didn't know very much. Now we're starting to get some evidence. This is work Ty Taylor, looking at the Amazon and just saying, okay, in general, this is all what would be one PFT, tropical rainforest. And if you go and look at this, what's the fraction of isoprene emitters in that forest at different temperatures? And he saw a significant increase in the fraction of isoprene emitters as temperature went up. And this and some other studies giving us kind of an inkling that it looks like you may be getting more isoprene emitters. They may do a little better, maybe. And there's some reasons, some ecological reasons that might be true, that isoprene as a temperature protection and could be a good strategy. It's still very uncertain, but something we know. The other thing is, a lot of these studies looking at land use would say, okay, we have a forest, lots of BVOC emissions, we cut it down, we put in a cropland, no BVOC emissions, or very, very tiny bit. A bit of an oversimplification, one we have to do when we're starting with very little knowledge. But in fact, as we look at this, and especially in developing countries, some of the tropical countries where a lot of that deforestation is taking place, this was a study in Kenya, Liu et al. We've looked at this in South America. And what you see is not necessarily a big change. So here you can see the isoprene concentrations from the forests that were there. And then you have the agricultural areas, which tend to leave a lot of the trees, and they're mixed agriculture. It's not going in and like Iowa and knocking the forest down and planting all corn. They're putting in a variety of things. And the isoprene's not changing so much. This isn't saying that this is always happening everywhere. We know it's not the case. But the idea that we should believe a forest going to a cropland is zeroing out emissions is also not the case. The other thing is that as we go with these general vegetation types changing, and including like going to the agriculture, is just, how many trees are there? In our models, the croplands will say no trees. We know it's not the case. And so in this study with, I collaborated with Weihua Chen and Shumei Wang, and looking at, can we take all the good tree data from ground studies and satellites and really see how our trees changing and what's the impact on isoprene. And of course, you look here, you see red areas where you have increases, although a lot of those show this is a percentage increase. A lot of those are in areas that didn't have many trees, so you don't need to add many to get a big percentage increase. Whereas some of these others down in the tropics, were a lot of trees, going down. So actually, when you average this out, even though there's more red than blue there, there's still a decrease. There's a global decrease in trees, and we estimate maybe something like a 1.5% per decade decrease still going on there, but not as high as what you might assume just taking these general types. And because the other thing that's looking at tree numbers, but we also have to consider what fraction of those trees are high emitters and not. And there have been estimates of that we used to have 5 trillion trees on the planet. We've maybe knocked down 2 million of those, down to 3 million trees. Other people have other estimates, but just going with this example here. So does that mean that we lost 40% of the isoprene? Well, it depends on whether those were those isoprene emitting trees or not. Some are, and some are not. And there are these programs now with the UN. The US was also pushing this. There was an executive order that we should plant 1 trillion trees around the world. I don't know if that will happen or not, but if we did plant 1 trillion trees, what's the impact? Are we half the way back to where we were in terms of isoprene emission? Maybe if the global average, if we're planting the global average of isoprene emitters, but there's some evidence that actually the trees that we tend to plant in plantations tend to be more likely to be isoprene emitters. They just do well in high light, more stressful environments, warmer temperatures. And so if, say, it were 70%, then we'd have about a 78% increase, much different than if it's just the number of trees. So we've got to be thinking about the amount of vegetation, but also the type of vegetation. And some indication that it'll be increasing more than what we would think. So to wrap this up with temperature then. So that was the CO2, the land cover, now the temperature. So there are 3 things that we're need to think about. And the first is the short-term variations. So the kind of the minutes to hours. This is something we think we know really quite well in general in the model. We can get this well, although Ned knows we haven't looked at this close enough, and there may be some really interesting things about high frequency fluctuations. So we're going to get that worked out, Ned. But in general, for the models tend to agree with the kind of hourly things. Then there's the past temperature, something we've known about for quite a while. Gabby did some wonderful work in looking at this. I don't know how long ago that was. It was a while ago. And it's, we have it in our models. Most models don't, though. So if you go back to the CMIP there, one or two out of all those models would actually incorporate something along these lines, even though it's still quite uncertain. But so we know there's that. And then extreme events, especially heat waves that we think could be happening more often. What are these going to do? So starting with the long-term flux record, if we kind of look at the behavior here, so what this is the longest BVOC flux record in the world by far out at UMBS. And so 5 years of data, and then there's 4 months there, June, July, August, and September across here. And then you're looking at the diurnal average for that month. And if you look at kind of the pattern of the curve, and that's why I say we can get that curve right, the pattern of it. We can do that pretty well, but you can see the magnitude, we're not doing so well. There's some huge differences here in the month-to-month, even within a year, and the year-to-year. And so B. Kiritana Nuvong, a PhD student working with me, has been looking at this data set. The data set's been out there for a while, but I think we're really gleaning, B is gleaning a lot more information from this. And so one thing is we just take the standard Megan model. Yeah, it's missing things. And also just the shape of this, the errors, doesn't look so good. So you can do things, machine learning analysis. You can get it to fit really well right here, like the PDF. So this pink right here, perfect. You can get the average right. You can get the two sides right. But you're still wrong. You're still quite a bit wrong in a number of those seasons. We can fit this, and presumably that also means if we take this to some other place, although we don't know because we don't have long-term data sets in other places, but presumably we would be wrong there too. So there's still things we need to learn about these seasonal to interannual variations, and I think temperature is a big driver in that. Another thing that this came out like 8 years ago for our colleagues. Rika Rienen and others at University of Copenhagen had this paper saying up in the Arctic, and they had some nice warming experiments where they had these little shelters out that warmed up the tundra there in Greenland a couple degrees, and they calculate a doubling for 1 degree change in temperature. It's like, wow, that's a lot, really unexpected. And then they did, in various Scandinavian sites, they did above canopy flux measurements and also saw this really high temperature sensitivity, far higher than what we have in the models, where a few degrees of temperature would make a big difference. Not quite a doubling for 1K, but much higher than what we have in the models. So we thought this is interesting, and so we, through the generosity of NSF polar programs, we had a consortium called Barons. This is a really nice logo that Hui Huang came up with. If you need a logo for your campaign, talk to Hui. But this was a collaboration between UCI, Kelly was at UC Riverside at that time, and Rebecca Sheasley at Baylor. But so our students from UCI, Hui Huang and Allison Welch, I had them out there at Tulik and doing enclosure measurements, so taking chambers where we could go out and control the environment. So we can keep light the same and control the temperature. We did some of these in the lab where we'd cut plants. We did some out in the field where we would take instruments out there to work on, and so we could see how things were changing. And then some of these studies were done out through the course of a season, so we could see as just the outside weather was changing, how was that changing the emissions. So that long-term effect and then this short-term effect. And then Allison was working particularly looking at shrubification. So the idea, our thought is like, yeah, what we have in the models, you get more shrubs, we'll have more isoprene. As it turns out, and I won't talk about this, but it wasn't the case, that actually more shrubs in there we think isn't going to be so much more isoprene. Not as big deal as we thought. And of course, if you go up there in the Arctic, you got to bring back, in the summertime, you got to bring back a picture of giant mosquitoes going over there to let you know how difficult it is to do this work here. But it's a really beautiful place. If you get the opportunity to go up to Toolik and spend some time there, Kelly's nodding her head, and it's just a wonderful setup that NSF has as a facility, and for NSF-supported research to go up there and do there. So this is one of the plants that we're interested in. It's about this high. It's a couple centimeters high. Anybody know what this is? It's a willow, not the kind of willows that we're going to see around here in Colorado. In fact, it's one of the smallest woody plants in the world. But it's a willow, so it's an isoprene emitter. It's emitting isoprene. But a really hard guy to get down there and get our enclosure around. We did do measurements on this, but mainly we worked on the little bit bigger willows that are around there. And so we did studies out there, these gas exchange systems, looked at what these things were doing. And so these curves are showing on here, temperature versus emissions, all normalized, so they come together at 1 at 30 degrees. And this green line here then is what we have in the model. This is what the model's expecting. These lines right here are what is observed from the above canopy flux towers running for months out in Scandinavia, right? So this is the kind of thing that they've been seeing, this really high temperature response. Here's what we saw with the willows for this. This is the short-term response. Over the hours, we change things. It's not so different. If anything, actually less of a temperature response. Clearly, this is not explaining that high temperature sensitivity that you see here in these curves. We did, however, see that past temperature. So if we look at the temperature of the past day, and here's what we put in the model, based on the work that Gabby and others did. We have this, what I would call kind of a conservative algorithm we put in there, and we don't want to make it too big, stick our necks out there too far. So kind of this conservative algorithm, but which still has quite a bit of change, 15% per degree sort of thing. But this is quite a bit higher than that. So much more sensitive in what we're seeing in these willows. And you can see that we really had nice conditions for kind of looking at this week to week. So we had it snowed and it was really cold, and then it warmed up and was really, really warm, up into the 20s, warm for there. And then cold again, and then warm again. So we really got to nicely see that happening. Wei put this together and into an algorithm and into a model, and you look and you see, yeah, this is a significant thing. You can maybe get a doubling kind of thing. This is important, but it doesn't explain what the Europeans were seeing. It's not enough to explain that. Part of the story, we think, but not all of the story. So to get the rest of the story, we went to this plant. Anybody know what that is? Oh, I forgot. No botanists and biologists in atmospheric chemistry. This is a sedge. So a sedge is kind of a grass-like plant, a little woodier. So most grasses don't emit isoprene, and we tend to think of the tundra as not a place where you're going to get isoprene. The sphagnum moss in the tundra is a small source, but these sedges are a big source, and there are a lot of them, and they emit a lot of isoprene. And so that's been known before. We went and studied these sedges, though, for their temperature sensitivity, and here's what we saw. So this, again, this green line here is that model that we're looking at, and then this line, kind of hard to see, this orange line right here is the sedge. So going right along with what we see with those above canopy flux measurements in Europe, and then Wei went in and looked there and saw that, actually the ones that had the strongest sensitivity were the ones that were mostly sedge dominated, and the ones that were shrub dominated, not so much. So really nicely kind of fell together that the sedges were the answer here. And the sedges are just behaving differently. So we went back to Irvine and got some local sedges and going in our growth chambers there. And actually, they look a lot like this Arctic sedge. Thought, yeah, maybe it's an Arctic thing. Seems to be this is what sedges do, which we don't understand why yet, but also to understand how many other plants do this, right? If it's just sedges, that's still important for the Arctic, because they dominate a large part of the Arctic. But we think that actually probably this is going on in other places too, and especially kind of grassy places that we tend to ignore from BVOC emissions. We just don't go look there. Maybe things like ferns are what we should be looking at, right, Monica? They may turn out to be the missing link that ties it all together. You knew it all along. You knew it all along. But so weigh again, put this in, new algorithm, put it into the model, and what the model would predict, we predict increases in the Arctic. It's warming. We've been predicting increases in emissions, something that would be something like 5% a year, quite a bit because it's warming a lot there. But then this is maybe a factor of 2 higher than that. And so this, combined with the two, what was going on with the willows, what was going to sedges, very different things, but seems to combine and explain a lot of that story. So the other thing is, well, what about these extreme events? Temperatures get really, really high. This is just kind of showing a standard plot. This is work Sanjeevi Nagalingam, a postdoc working in my group, has been working a lot looking at heat. How does heat stress affect these emissions? And this just kind of shows you the standard sort of thing that we'll see. For some compounds from some plants, what you'll see, and here we're going from 30C here, this is 40C, 43C, then back down to 30C. And so the standard thing you'll see with isoprene from all plants, and some monoterpenes from some plants will follow this. And these are compounds that are just being produced. They're not being stored in the plant, they're just being produced. So what happens at high temperatures, those enzymes just stop working. The machinery shuts down. And so they just can't keep going. They can't keep photosynthesizing either. But even the short term, the enzymes will break down first and they'll just stop doing it. But then you can see a curve like this, which just keeps going up and up and up and up and up until you cool it down. And these, we think, are monoterpenes stored in the plants, and plants have all kinds of different specialized structures where they make this for the life of the leaf and they store it there, and there's plenty to get emitted. So looking at these two behaviors, and this is just showing, so different compounds here across the x-axis, then 18 different tree species across the y-axis, and you see a lot of different things. The point of this is, so different compounds from different plants, different compounds in the same plant, different plants, same compound will be doing different things. And what we're seeing here, the reds they increase a lot, like 100-fold or more increases with heat stress. Some of them decrease with heat stress. So we have these different things going on. Some of them not so much of a change. And some examples here of that data, and looking here at a sunflower on top, a needle leaf tree, a cedar tree in the middle, and then a broadleaf sweet gum at the bottom. Each case here, the model is going to predict around a factor of 4 increase in emission. But what we're seeing is an increase of factor 22, 98, 5,900 kinds of increase. And so some, a really large increase, and some just an incredibly large increase that really changes things. And it's not just during those moments of increase. So here we're looking at, well, what happens afterwards? So you've done this. And so in some plants, in this case, we're looking at 2 close cousins in the same family. So there's a white cedar and the Chamaecyparis genus, a juniper in the Juniperus genus. So they're both needle leaf, look pretty similar kinds of trees, but still have somewhat different storage structures. So even down on the genus level, species level, you can have different ways that they store these things, but also some broad generalities that we hope will help us with modeling this. But then what we see, so these increases, so really strong increases, this case, like 3,000-fold. And basically, you're kind of going from just nothing, from barely just being able to detect this, would have a real small atmospheric impact, right? This plant is maybe making PPT levels out in the environment to really strong emissions, as strong as our strongest emitters. And then you cool it back down. It just starts dropping down, right? So here it's dropping down, and but still elevated for some compounds. But with others, like the juniper here, you turn the heat off, and it just keeps it. In this case, actually, just continuing the heat. It just keeps going up and up and up. But when you turn the heat off, you'll have elevated emissions can persist for a week or more. So indicating like a heat stress event is not just going to be that day of the heat stress. Maybe they last a day or two or three or something. But these emissions are going to be going on for maybe a week or two. And if it's 1,000-fold or more higher, this is going to be the most of the annual emission for this if you're having heat stress events. Also, since chemistry is very nonlinear, it matters that all this stuff is coming in this one day. You could have a really big impact on that day rather than a very tiny impact over a long time. So going back to this, what can we say about this? So with CO2, I've said various models would say about -3% to 6% a decade. And it looks like it should be on the low end of that. 3%, and probably as we incorporate this temperature effect, even less. The temperature effect, 2 to 7%, looks like probably on the high end of that. Still a lot of uncertainties, especially we've got to figure out this heat stress stuff, which is going to be difficult to really work that out and quantify it. We need to be seeing it out in the real world, long-term measurement studies to really figure that out. And then the land use, land cover change, some interesting things going on. I think there's some indication that this is going to cause an increase, but we really don't. This is still in the level of uncertainties that we can't say too much. But I think given all that, we can say this is the idea that emissions are not going to change in the future, I would say are not implausible, and actually looks very likely that there are changes and could be very large changes, particularly in certain regions. The global total probably doesn't matter so much. What's going to matter to people is what's happening locally and how this is impacting local air quality and climate. I think some of the reasons for optimism that we can start working some of these things out include things like the satellite observations that we have. We can already see this, fortunately, with satellites like OMI out there giving us these long-term data sets. And it's getting better. With the new satellites, with having geostationary satellites are going to be a huge boon. But of course, it's not this is going to help us a lot with isoprene. It's not all compounds. It's not going to answer everything. But what we see already, just from the older data sets, this is a 2005 to 2013 data looking at the isoprene trend percent per year. Remember, before all those other things, I was saying, well, on the high side, maybe 8% per decade. So that's 1% per year. What we've been seeing in the Arctic is 10% per year going on. That's what we get in the model because it's been warming so much there. But it's also what you would get from the formaldehyde data in OMI. But the satellites aren't going to answer everything. We really need more measurements. We need to understand these processes better, but we need long-term in situ. Yeah, I didn't put the word long-term in there. We need in situ data. The long-term flux measurements are really key to figuring this out. So kind of tower data that we can do, but also aircraft flux measurements for giving this idea of how things are going to change. For that land cover change part, the aircraft is a wonderful tool for going out and seeing really on the ecosystem scale what's going on. We can scale up, bottom up, but we don't have a lot of confidence in what that's getting us. So I'm going to end with this. Thank you all for being here. I'll be happy to take questions.
Thanks. Hi, Alex. Excellent talk. Thanks a lot. Justin just showed the trend series from
The satellite?
the satellites,
Yep.
yeah, from Megan and from OMI. So what are the major factors that drive the trend? Is that the land use change, the temperature? Does it depend on region?
Yeah, no, it's a good question. So in Megan, then here it's temperature. In the real world, I don't know, how much these other things are happening. We wouldn't think that, there is land cover change going on there. There's shrubification going on. From the data that we have, although it's just at one point at Tulik, would suggest that that's not such a big deal, and it shouldn't be happening at this rate. So we'd think it would be temperature.
Thanks.
Yeah.
Thanks, Alex. Great talk. I have two questions. Could part of this be due to methane?
The part of which?
This.
This right here, in terms of what you see in the satellite?
Yeah, or you get formaldehyde from methane. Right?
Yeah. Well, we should talk with the satellite
So
people.
that's why that was question
Peter
one.
Nell
Question.
might have an answer for that. I have no idea.
Question two. So when you bake these plants and you say it kind of keeps going for several days after that, is that a destruction of plant tissue and it bleeds out and dies, or what happens to it after this treatment?
Yeah, we think so. We've been interested in work with a colleague at Cal State Northridge, Chandak. We can look at gene upregulation, right? So there are two possibilities that are going on. One, you're upregulating genes, right, that are stress genes, and we know that happens, and we think that's some of that. The other is the physical process. You had this thing all cased in, and now it's not. It broke, and it's coming out. It looks to me like more that latter seems to be the dominant thing. We really haven't seen so much the gene regulation.
This would kill the tree.
Well, not necessarily. Not necessarily. No. I mean, it's not gonna be photosynthesizing for a while, but it won't kill enough of it. Will kill a tree, but just knock it back a bit. And trees have a lot of defenses to keep going, and then they'll come back. The weeds in your garden, right? You can't get rid of them. But it's a really good question, and it's going to depend on the species. So what you're going to see is, some trees they can't handle it. They're going to be out of here. But other trees, are kind of the cockroaches of the botanical world. They're just really have good strategies, for dealing with it.
Hi, Alex. Very nice talk. So a lot of the responses of the isoprene emissions and monotropin emissions to environmental conditions like CO2 and temperature seems to closely tied to the stomata activities for the plants. So is there any mechanistic model or scheme that has been developed for model use for this? And related to that, another issue that you haven't mentioned in this talk is the water stress to the plants. So there's actually some study suggesting that if plants are exposed to drought conditions, then it will also change the BVOC emissions. So can you comment a little bit on that? Thank you.
Yeah, so as far as the stomatal conductance, doesn't have a direct impact on most VOC emissions. There are exceptions with some of the water-soluble compounds like methanol. Methanol is coming out in the water stream, and it's really tied to the stomata. Isoprene, which does come out of the stomata because it's produced, and that's where it gets out. But you can close the stomata with respect to CO2 and water, and the isoprene still gets out, because it builds up ppm levels in there, and it's a gradient thing. The resistance is high, but it's such a big gradient that it still gets out. But it doesn't mean the stomata aren't important. So one thing that's certainly important, it's so temperature sensitive that the stomata controls temperature, right? So with transpiration, really important for that. So getting leaf temperature right means you need to get the stomata right. And so as far as whether models do that, and so we have Megan and CLM, then we rely on CLM to do a good job there. And it does have that. It's not perfect, right? And I think the model errors and leaf temperature are important. If we could improve that, it would improve these estimates. But certainly in models like that, those with some dynamic vegetation models or those kind of land surface models that have stomata conductance, it's in there and helped. But also the other thing I'd say with drought, and that explains a lot of the drought effect. So we see with mild drought, you get a big increase in isoprene. And leaf temperature can explain a lot of that, but I think not all of it. There's also studies where we take these enclosures and you measure at a prescribed temperature, right? So you measure 30 degrees, and then you drought the plant, and the emissions change. So it's not just the leaf temperature driven by stomata. There's other changes going on in the plant, which is not surprising. Most of these BVOC emissions are related to stress. And some of them are maybe some direct effects, protecting the plant, stabilizing membranes, beating off all the pests. But more and more, biologists, ecologists, biochemists studying this are thinking that a lot of this is about signaling. That a lot of these VOC are signaling, and it's a way for the plant to signal the whole plant to turn on. It's like, hey, we've got the roots saying, hey, we've got a drought coming on. Otherwise, how does this leaf going to respond? And so there's things that we're gaining an understanding about. Yeah, good question. Thanks.
Okay, Peter now.
Thank you. Very good talk. So I'm not really in the plant world. Most plants in my house die, so that's my knowledge.
Oh, you're doing good stress studies. We should be measuring the BVOC from those.
Might do that, yeah. So many of your examples, and the one I have now in my mind is the one of the trees, with this very, very different level of response to heat stress. So it must be a huge amount of plants which you have to categorize to get. So is there a way to do that? Can you say that similar type of trees will have a similar type of, because it's, the example you gave on the north, the polar program with this other plant, which I forget the name, with the white things, that then explained the isoprene. It looks like to me, well, there might have been 100 other plants on that area, and how do you do that?
So the hope would be that there would be some pattern that then could be related to PFTs or in global models or something along those lines. And so Sanjeevi's been working at this. The strategy, the plants that he's choosing is we go and look and trying to look at these broad classes of structures. Some have these external trichomes where they have these hairs, basically. I've seen plants that have hairs, and at the end of that hair, there's a little terpene storage vacuole. That stuff, you touch the plant, and it's going out. Others are glands right on the surface, and the stuff's coming out, or resin canals. Others are buried down in the plant, so they're down below the epidermis, right? And so with that, you got to break something or do something. And so kind of been looking at these different categories with the great, we'll see this, and each category will be completely different. And it's as things work out, it's not exactly like that. I think we can see some things. I think it's not going to be easy. I think it's going to be difficult. But then, there's certainly things we can do, having some kind of average effect. Certainly better than ignoring this and not doing it at all. But yeah, it's such incredible diversity and complexity in the biological world. We're going to have a hard time really nailing this down.
Okay.
Maybe we can first ask some question online. There's a previous one from Doug Worsnell. Okay. Any work showing ecosystem level increases in monoterpene emissions associated with insect infestation, such as beetle outbreaks in the Rockies?
Yeah, so there are studies, and including things where we were doing when I was here at NCAR. That was one of our interests, and we looked at that. And so you certainly can go out, put enclosures around branches, put them onto the trunks, and look at this. A number of groups have looked at this, and and they'll increase as you expect, right? And so you're breaking all these storage structures. It's coming out. People have done modeling studies suggest that this can have some impact. But it kind of gets back to this and what Peter Nell was saying. To really have this in the models. There have been efforts to put it into models and kind of prescribe it in some empirical way. We could say, yeah, there's an outbreak, there should be increase in emissions, but we certainly don't understand it very well. We don't have any kind of a mechanistic description of this, but we can say, yeah, it should increase a lot.
Question from Doug Worsnott. Your talk suggests that the uncertainties in BVOC trends are somewhat equivalent to those in aerosols, especially since non-isoprene BVOCs likely have a disproportionate impacts on aerosol. Very good talk.
All right, thanks, Doug.
Any more questions? Yes, okay.
A very nice talk, thank you.
Thanks.
I was just staring at this slide because it was there too long. So I have a quick question. What's going on in the Southeast US? Why do we have an opposite sign? And did we find out the reasons, given that there were several campaigns there?
Yeah, good question. So, and I think one of the starting points is how good is this OMI, right? I mean, and then the other one, Megan. So we know Megan's wrong, has a lot of things wrong in it. No question about that. I know that for sure. As we compare it with the satellite data, though, kind of got to start with, well, like how much confidence do we have in that to do these kinds of things? I mean, my feeling on it is that this is getting great. This is getting, 20 years ago, started looking at these formaldehyde things. It was kind of like, now why is it one day, it's showing a lot of isoprene, the next day with the same conditions, there's no isoprene? Yeah, I'm, not so sure about this. But things are improving a lot, right? It's getting a lot better. And with the new satellites and things, I have a lot of confidence that it really will be a constraint, and we'll see. But yeah, right now others could answer that part. As far as what say is going on, so that's showing a decrease there. I mean, that can't be land cover in that amount of time. It's not CO2, but could be some temperature effect going on. But the model, Megan should be driven by weather data that's reasonable. So I don't know. Maybe it's methane.
A question from people online. Could the so-called greening of the planet be counterproductive for the future of the planet?
Well, so one thing to keep in mind with these BVOC is that if we keep the air clean in a place that has clean air, these things are not making ozone and particles, right? So let's not blame the trees. On the other hand, I think we're at the point in urban areas where we're trying to get ozone and particles down. It's such a struggle. We should be thinking about the trees and planting trees with low BVOC because it's not natural anyway, right? We green our areas in cities, in plantations. We should think about it. We should not be thinking about it for natural landscapes, natural ecosystems. We have to protect biodiversity. There should be no touching the isoprene trees in the natural environment. But in the artificial environment, as we're greening our artificial environment, let's think about these BVOCs.
Okay. Okay, yeah.
Thanks, Alex. That was great.
Thanks,
Curious about your UMBS example, and how do you know that it's Megan that's failing and not the
Not the measurements?
And, CLM. It looks like there just aren't leaves on the trees in the model. I mean, or that there are leaves when there shouldn't be on the deciduous plants or something like that.
With UMBS?
Yep.
Well, yeah. You're right. But this is actually driven, this is not CLM. So this is driven by observed LAI.
Okay. Not sure I understand that.
Well, just so you know, LAI was measured and we, or actually we use satellite. We use satellite LAI to drive it.
Okay,
So this is not CLM driven. And if it was CLM driven, and we've seen that, I mean, you know, if you just kind of grab it and throw it out there, it'll be really off, right? And then you do it and you do all the things you're supposed to do with it, and it gets better. But we've done that in particular, like looking at the Missouri study, we're looking at drought, you know, and looking at CLM. If you really got to get it tuned right to work. But in this case, this is being driven by observations. The inputs are all observations, which have uncertainties and errors. But.
One more question from online. Is tree age affect the emission, for example, secondary forest?
Yeah, it's an interesting question. There's evidence that tree age is important. You know, one thing is eucalyptus trees. We've always considered those as really high emitters, and that's kind of based on getting, you know, eucalyptus plants growing in people's labs in Europe and the U.S., and they have really high emissions. Turns out a lot of the mature eucalyptus trees, the emissions aren't so high. And so we know that is something important. It can kind of go in the different direction. It's not exactly clear. We don't have that very well worked out.
Okay, in the interest of time, maybe we can take one more if we have. Okay.
Yeah, I agree with Doug there. With Omi and Megan there, that seems a little too much to me. It's not my result.
Well, I agree with Doug. Also, I think that comparison is actually quite good. So for the Omi formaldehyde,
good.
it was how many years was that image?
How reliable do you think that is, Peter? Now, the trend that you would get from this. The other thing is
2013. So
the other thing, keep in mind when you go from Omi and get an isoprene emission, you're also using a model.
yes,
Right? We use a bottom-up model for Megan that includes satellite observations of land cover. With Omi, you're using a satellite observations of formaldehyde going down to emission, but both are models. And part of this may be those models have some similarities that cause this to happen.
Yeah, so you have a lot of steps from the measurements to isoprene, basically.
Yeah.
And formaldehyde is a challenging product to get out. And I see that the time range is okay, I think, for the instrument, so not after 2014, 15. I think maybe it should be repeated with Tropomi, because those data are much, much better.
Yep.
We have now 7 years, so you can
Yeah,
do
I've really been
something.
impressed when I look at Tropomi and things like and including with things that look like in the U.S. and people have used Omi and like in Texas and say, well, wait, Megan's wrong. It's like, yeah, we know Megan's wrong, but you know, there's this thing here. Then you look at Tropomi and it's agreeing really well, which doesn't make it right, but it's encouraging to me that a lot of these things that you know seemed kind of wrong. You know, we're kind of like, well, I don't think we'll change it just yet because Omi came up with this. But Tropomi seems to be saying, oh, actually, it was good to kind of wait to try to update that.
We have a higher sensitivity in that instrument.
Yeah.
And you have a higher space
It's really
resolution,
impressive.
So it's double impact, basically. And so I think that would be the next step. Because in Omi, a lot of the formaldehyde data, you know, it's 100% error, basically.
Yeah.
And then this
But you're right, it'd
is-
be really interesting to do this again.
Yeah.
And of course, with this, I was just a support person, and this is the Belgian group doing this.
Yeah, they're
this
very
work.
good. So this product is
Yeah.
good. Yeah.
Yeah.
This is the best you can
But
get.
it should be done with Tropomi now.
Okay, one last quick.
One more comment is that there are now direct observations of isoprene from Chris, and those could be used to also check the assumptions in the model that you go from formaldehyde to isoprene.
Yeah, and they seem to be really complementary, which is great, right? I think either one, so they really both support each other and combining those two. Yeah.
All right, thank you everyone for attending, and thank you, Alex, for the great talk. And
Yeah,
we'll
thanks
see
for
you in
having me.
March.